Gracie. I'm based in Duke NGS actually, and my supervisor is David Workshop. So I am a molecular biologist by training now. So I'm guilty of the jargons that David mentioned. So I will try my best to go through them as clearly as possible, but feel free to ask me any questions if you don't follow whatever I'm saying. So today I'll be talking about circadian rhythms, which is essentially our body cycle, our body clock, and some ways of regulating it. So I'm sure a lot of us would have, experienced jet lag at some point of time or another when we travel from one time zone to the next. So for the first few days, our clock sticks to the time of your originating country, and then you're cranky, you're tired, and you don't understand what people are saying very well. And then it takes a few days for your body to adapt to the new time, and then you're functioning normally again. So this is essentially our circadian rhythms. Here I'm showing you the time lapse of a sunflower. So we know that the sunflower follows the sun. So you can see that this flower follows the sun over the course of the day, and then it rests at night. The coolest thing is the plant is able to position itself even before the sun rises, so that when the sun comes out like now, it is already in the prime position to capture as much sunlight as possible. This tells us that the plant inherently has a clock, which dictates what time not the time, but dictates when it should be positioning itself to capture the sun. So what exactly is the circadian rhythm? It is essentially a roughly 24-hour clock in our body, and it regulates the biochemical, physiological, and behavioral processes in our body. So our main clock lies within the brain in the higher organisms, and this clock is synchronized to the external time by light. And the main clock actually synchronizes all the clocks in all the cells in the body so that your entire body runs on the same time. However, in the absence of signal from the brain, the cells are actually able to maintain their own 24-hour cycle. So why is the circadian rhythms important? Well, circadian rhythms have been associated with diseases such as heart diseases, diabetes, obesity. It is a factor in drug responses, and recently it has to be found and to be important in cancer. So with all this in mind, it's getting to be important to understand how circadian rhythms is regulated so that all these diseases, especially drug responses, can be managed better. So I'm going to go a little bit in depth now. So at the molecular level, there are two proteins that are very important in the regulation of rhythms. So early in the day, transcription factors will turn on the transcription of two proteins known as period and cryptochrome. So fur and cry they work as dimers to inhibit their own transcription, which is synthesis of the protein. So this is one of the process, and then their level will peak in the early night. So another process that is important is another protein known as Casein Kinase 1. Kinase is essentially an enzyme that phosphorylates another protein. Phosphorylation meaning they put a phosphate group on some other protein. Okay. So CK1 phosphorylates PER, and this phosphorylation will cause PER to be targeted for degradation. So if you imagine a machine gun with a targeting light, if you target the light at an enemy, the aircraft will come and attack that enemy. So, anyone watch Transformers? Right. So with that degradation, the level of PER will reach a minimum somewhere in the early day. And then because PER is now gone, the transcription repression is no longer there. And then the synthesis of PER will start all over again, and the protein level will rise again. So with that, you can see that the PER level actually oscillates over the course of the day to give you a very nice wave over 24 hours. So in our lab, we have a system to measure rhythms we have cells that carries PER2 protein fused to luciferase. So luciferase is an enzyme that converts luciferin to a light output. And this, when we put it into a machine known as the lumicycle, the lumicycle essentially is a reader. It measures light intensity in live cells in an incubator over the course of however long you put it inside. Then this data can be analyzed using the software. So this is an actual output from the machine, looking at the light levels over time. And because PER2 is fused to the luciferase, the light output is equivalent to it's representative of the PER2 levels. And you can see that the peak to peak in all this, each peak to the next peak is 24 hours, which is exactly 1 day. So the phosphorylation of PER by CK1 is actually a key event in circadian rhythm. Why do I say that? Well, several evidence have been found that CK1 is important, and one of it is the discovery of a mutant known as tau. So CK1 epsilon tau is a short period mutant. It was first found in hamsters. Why do I say it's a short period mutant? If I put this cells in a lumicycle, you will see that the peak to peak is every 20 hours. So it peaks at 20 and then peaks again at 40. So these hamsters operate on a much shorter day than the rest of us. So that's the first evidence that CK1 is important. And the next evidence is the inhibition of CK1. If I put my cells in the lumicycle and I measure the period using the peak-to-peak analysis, wild-type cells, as in cells with no treatment whatsoever, they oscillate at a time of 23.5 hours. If I inhibit only CK1 epsilon, which is one isoform of the kinase, there is a very slight lengthening of the period to 24 hours, which isn't so significant. But what is significant is if I inhibit both isoforms delta and epsilon with another drug, the period is lengthened significantly to about 27 hours. And this shows that CK1 function in the rhythms is very important to maintain it at the 24-hour cycle. So the regulation of this CK1 phosphorylation of PER can be by two ways. One is by regulation of the kinase activity itself. That part I will not be talking about today. And the other way that the phosphorylation may be regulated is the interaction between kinase and PER2, which is what I'll be showing now. PER2 is actually a huge protein. It's about 1,257 amino acids. And then it contains a lot of distinct domains, as you can see in this figure here. And most importantly, this region here is what we call the phosphodigon. So the phosphodigon is actually the part of the protein that is phosphorylated in order to be targeted for degradation. So it's where the machine gun light is pointing at. Then the region I am most interested in is this region over here, which is the CK1 binding domain, because this is where CK1 interacts with PER. And the two regions in blue I showed here, previous work have already found that deletion of these two areas cause CK1 to be no longer able to bind to PER2. Then this region FXXF have already been characterized by other groups, so my work focused mainly on here. So in a model, for you to see clearer, if you imagine PER to be a bent protein like this, CK1 is interacting with it at two different areas, which are the blue boxes. If I deleted the two boxes, CK1 is no longer able to bind. So this brings me to my first question of whether there are any residues in this region that are important. And I did a sequence alignment of this region, and you can see that among the different animals, there's actually a lot of, the stars got shifted, but yes, there's actually a lot of sequence alignment over here. And the one residue that we found to be most important is T, uranium 605 over here. I will not be showing you the data from all the mutagenesis, I will just focus on that one residue. So what I did was I used a minimum binding fragment of PER. So this fragment only has the CK1 binding region and the phosphodigon. And if you imagine CK1 and PER to be in a complex in solution, if you use an antibody that recognizes PER and you pull the entire complex down with a E, you can see whether CK1 comes down together with PER. So in a Western blot, in wild-type PER, so this is wild-type, PER is pulled down and CK1 epsilon comes down alongside it. when I mutate T605 to alanine, PER still comes down fine, but you can see that CK1 epsilon no longer exists in the same complex. And then when I mutate it to E, CK1 comes down and PER again. You can see what is interesting is the PER2 level is higher when it's mutated to A, and this is actually not surprising because I have already said earlier that when PER is phosphorylated, it gets degraded. If CK1 can't bind to PER, it makes sense that it can't phosphorylate it and it doesn't get degraded. So this data shows that T605A mutation abolishes the binding between PER and CK1. Then next, I wanted to know whether this interaction is dependent on the kinase's activity. So remember my inhibitor that I used earlier. So I'm using like the same inhibitor that inhibits both isoforms of CK1, and I found that the wild type and the E mutant are still able to bind to PER2 in the presence of this inhibitor, showing that this interaction is independent of the kinase activity. So building on the model, CK1 interacts with PER at this domain as well as the T605. If I mutate the threonine to alanine, CK1 is now no longer able to bind. So if PER cannot interact with CK1, can it still get phosphorylated? So in our lab, this is a reagent developed by one of my postdocs. We have an antibody that recognizes PER that is phosphorylated at serine 478. Serine 478 falls within the phosphodiagon. So in the Western blot, the wild type PER can be phosphorylated at serine 478 just fine. The A mutant, although expression is equivalent, there is no longer phosphorylation at the serine 478 level, showing that when CK1 can't bind to PER, it cannot phosphorylate it, which makes a lot of sense. So in a cartoon way, CK1 binds to threonine 605 and phosphorylates the protein. And one of the phosphorylation site is the phosphodiagon. If I mutate this to alanine, CK1 can't bind, PER2 is no longer phosphorylated. So my last question is whether the protein stability will be affected. So I've already said earlier that the protein must be targeted for degradation by phosphorylation. And to understand this, you need to know that in the protein, the amount of PER2 is actually a balance between two processes. It's a balance between the synthesis of the protein as well as the degradation of the protein. So if I add a drug that turns off synthesis, it's equivalent to turning off the tap. If I turn off the tap, but the water is still flowing out, over time the water level will just be gone. So with this in mind, I did an experiment where I added that drug and then I followed the amount of PER2 in the cells over 4 hours. As you can see, in the wild type PER2, by 4 hours there's barely any protein left because they're all being degraded. But when I mutate the T605 to alanine, the protein is stable over the course of 4 hours, showing that the protein is a lot more stable, and this is the quantification of it. So PER2 that cannot interact with CK1 cannot be phosphorylated and is stabilized. So this is the final. That was in the fragment. So now I want to know the full-length protein. However, the full-length protein is huge, it's hard to work with. So what this postdoc in my lab did was she found that we can use the Lumi cycle to measure the half-life of the protein, because if you see the luciferase activity cycling, the moment you add cyclohexamide, the amount of protein will just drop to a bare minimum and so forth. So the data from this point to here can be used to calculate the half-life of the protein, meaning the rate is degraded. So in the full-length protein, while the wild-type PER has a half-life of about 4 hours, the 605A mutant has a half-life of close to 7 hours, showing that it's a lot more stable compared to the wild-type, and this is in agreement with the data I got from the fragment. So in the final model, CK1 binds to PER2 via T605, and that causes phosphorylation of the protein. So with the phosphodiagon phosphorylated, now the E3 ligase adapter, which is essentially the first protein to bind to PER2 to bring it to the machine gun. So this leads to the ubiquitination and degradation of the protein. If CK1 can't bind when I mutate it to alanine, the phosphorylation does not occur, the adapter can't bind, and the protein is not degraded. So all this is well and nice, but what is its effect in rhythms? So that is the big question. We want to look at the effect of T605A on rhythms. So that is actually work in progress because we can do transfection studies for that. So we are planning to mutate the endogenous PER2 in our mouse embryonic fibroblasts using zinc finger nuclease technology, and then look at the change in rhythms in these cells. So this is my lab. That's David Bersham, my supervisor. So Min here did a lot of the work because we are working very closely, and I thank you for your attention. I'm happy to take any questions.
What happens to purr and cry when we get jet lag? I mean, how does the body reset this whole cycle?
It's actually a regulation of the kinase activity. So if you imagine us living completely in the dark, we will actually stick to the original time. But when we are exposed to sunlight at a new place, in the new country you're in, the sunlight will actually activate your photoreceptor, which will send signals to your brain, and then your brain being the main clock will synchronize your whole body. But it can't do it at, say, 5 hours at a time. So you will find yourself being corrected like maybe 2 hours a day. So you need about 3 or 4 days before you get to the new time.
So if I were blind, would I still be affected by jet lag?
If you were blind, probably not. Although actually it has been shown that temperature might be a factor.
You are using a cell line that uses it in incubator, and it's a constant live temperature, everything is constant. You still observe the circadian rhythms?
Yes, because this is the standard 24-hour circadian rhythm. It's equivalent to putting animals in the dark. They will maintain their original 24-hour clock because in the cells there's no photoreceptors, so light is no longer a factor. So it's definitely what we call a dark cycle. It doesn't get entrained to environmental factors anymore. It's a very constant 24-hour cycle unless there's a mutation inside that shortens the cycle to say 20 hours in the counter.
Is there any way to reduce the effect of jet lag?
Well, I guess that would be the big main goal of my study, to see whether there's any way I can modulate the either the kinase activity or the kinase substrate interaction, so that, well, we may find something that helps with jet lag, or we may find something, because studies have been shown that if you give drugs at different times of the day, it actually affects the efficacy of the drug. So that's another goal of my research, that we can find the perfect timing, or we can give a cold drug to help improve the efficacy of the whatever drugs are taking.
In terms of doing a complete mutation,
That
would actually include people
correct.
approximation of your
Yeah.
I don't think cell lines approximate very well because it's very change over 24 hours. It's not mimicked by the cell line completely, only looking at degradation of period.
But we wanted to look at, to see the change maybe in the cell line first before we move on to the mouse studies. Yeah, but mouse will be better because we can look at behavior, we can look at how the mouse activity change over 24 hours.
Natural mutants of period that do not TCK1 phosphorylation.
There is another mutant. It's what they call familiar advanced shift mutant or something. These people is actually true people. They are morning larks. They wake up early, they sleep early. Yeah, so there is actually a natural mutation.
The pilots, people will travel all the time, crossing many time zones. How do they manage? Have they gotten used to it? Is there some other clocks in the body that is resisting to?
Well, another hypothesis is food actually can reset our clock. So, yes, my boss believed that if you don't eat before a big flight, and then you eat only at the time of breakfast, so after a long period of fasting, your first meal, your body will take it next morning. That's what my supervisor believed. That's still all there's still work to be done, but maybe that's how the pilot do it.
In terms of synchronization, when you measure the output from the substrate, is that averaged over the whole dish?
It is. So it's
So do all the cells, are they all exactly synchronous? Or do
we
they-
have to synchronize them before we put them into the Lumi cycle. And then over time, they will actually desynchronize, which is why the amplitude will get put down. Yeah.
And do they talk to each other versus two-dimensional culture?
Well, I haven't tried and see whether they talk to each other. But what others have done is they actually tried single cell synthase, the Lumi cycle in single cell conditions. And then all cells in the 96 well are running at the independent time of each other.
Thanks, Gracie. Your boss ever needs a test subject to travel around the world to reset the record by eating Army Man.
Alright, so right now let's go for a 20-minute short break and we'll convene at 11 o'clock for another round of interesting talks. See you later.